So I'm going to go ahead and get started and welcome you to the Vaccines Working Group session. And just to thank you, can you guys hear me? And just to thank you all for being here. It's really exciting, actually. We had so many people come, and I think it really reflects the excitement in the field. And of course, thanks to our organizers. So I'm going to go ahead and open the session. They all know me. I'm Dave Lewison. I'm the chair of the New Vaccines Working Group. I'm the chair of the New Vaccines Working Group and super happy to be here.
Thanks, Dave. It's not. Okay, hi everybody. I'm Jennifer Woolley. I'm Secretariat for the Working Group on New TB Vaccines, and I'll just echo Dave's thanks for being here. We appreciate that you took the time to come and learn about what's exciting and happening in the space of new TB tools. So we'll be talking about vaccines for the next 90 minutes. I'm just going to give a very quick update on the Working Group on New TB Vaccines. Many of you know us already. We are an informal network of stakeholders who are interested in, involved in the research, development, and implementation of new TB vaccines. The group is open to anyone who is supportive and interested in new TB vaccines to join, and we provide a platform for communications, for scientific exchange, for advocacy, and sharing information on new TB vaccines. We have several priority areas. One of those is advocacy. We work in coalition with the TB Vaccine Advocacy Roadmap, which is a coalition of stakeholders who do global advocacy and policy work around TB vaccines. We coordinate with them for global advocacy efforts. We place a strong emphasis on research literacy. We developed with the other new tools working groups a curriculum, an online curriculum on TB research to help build research literacy, and we'll be working on other opportunities to do that. We also place an emphasis on advocacy capacity, and particularly for researchers. To build the advocacy capacity, researchers have a critical voice in advocacy, and we want to help support them to be involved in that field. And linking these, we try to also build a bridge between the research community and the advocacy community. Another priority area is sharing knowledge and fostering discussion. We do that through our website. We have a number of resources, publications, upcoming opportunities, as well as information on the working group on our website. We compile a global pipeline of clinical and preclinical TB vaccine candidates. That's also available on our website. We're planning a series of online workshops and webinars over the next several months, and some of those will be open to anyone who wants to join. Some may be smaller amongst our membership, but there'll be a number of topics that we'll be discussing. So stay tuned, and we'll be sharing information about those through various platforms. And we have an online community that I will be talking about in a few minutes. We also recognize that the product development pathway is complicated and challenging, so we try to provide support for researchers who are working on products and doing research. We provide a dedicated webpage with resources on product development and help steer researchers towards services that are available to them as they are navigating that pathway. And I'm going to turn it over to Rebecca Clark from our ECR Network Steering Committee, and she's going to tell you a little bit about the ECR network, and then I will come back to wrap up this thing.
Hi everyone, thank you Jennifer. My name is Rebecca. I am an assistant professor at the London School, and I'm here on behalf of the ECR network, particularly on behalf of Carly and Paul, who are co-directors of this steering. And so who are we? Essentially, we're an informal group that connects and empowers ECRs in TB vaccine research, offering research, resources, support, and networking opportunities, and fostering collaboration, knowledge sharing, and professional growth. And it's led by our ECR-driven steering committee, which is a group of about 6 or 7 of us ECRs from across the globe who come together. And so in 2005, so far what we've had is our online community launch in November. And this has been a great opportunity to bring ECRs together in our interactive conversational engagement. We're able to mix technical insights and personal growth topics, and just to have discussions amongst ECRs about the TB vaccine research that we're doing. We also have an exciting event coming up, which is the virtual coffee corner event. And so this is more just an online networking opportunity for ECRs and TB vaccine research to have discussions about everything that's going on in our research and otherwise lives. So moving forward to 2026, we're really looking forward to building momentum in the online community. We're planning to have virtual sessions for ECR network members. And so this will involve informal discussions among ECRs, and then also networking sessions with ECR supporters. So getting some support for ECRs from other members who maybe have a bit more experience and have their experience to be able to offer. So a couple links here. If you'd like to learn more about the ECR network, or if you know someone who is an ECR and would be interested, please follow the QR code there and learn more about the network. And then also for anyone interested who is an ECR in attending the 4th of December Coffee Corner, please register for a link to sign up, and we look forward to seeing you there. And thank you very much.
So Rebecca and I have both mentioned that we have an online community. We just launched it, literally just launched it about a week and a half ago. This is an online community platform. It's brand new, but it's going to allow real-time engagement among working group members and TB vaccine advocacy roadmap members from around the globe. You can find and connect with others who have similar interests or who from the same country or region. We're also using it, I said we were trying to build bridges between advocates and researchers, and also with ECRs. People can identify themselves as an ECR or someone who wants to support ECRs, or if you're willing to be an advocacy mentor or work with advocates on research literacy, you can identify and find each other there. There'll be online discussions on numerous topics. These may happen in written posts or on online sessions where we can connect with each other. We have dedicated areas for early career researchers, for advocacy, and for all topics related to TB vaccine R&D in our space. And it's open to all members of the working group and TB Vax-Arm. So I encourage you to, if you're already a member, to become involved in the community. If you're not, to join us, and you don't have to know. Just try to join the community, and we'll tell you if you're a member and we need anything more from you. But we hope to build this community. As I said, it's new. We're really excited about it, and we hope you'll be involved. So this is just a wrap-up slide. There's ways you can get involved with us through our website, becoming a member, joining our community, subscribing to our email updates on social media, and you can always get in touch with us with any questions you have. And this is all available on our website. So with that, I will wrap up and just want to acknowledge and say that we're really grateful to all of the individuals and organizations that provide their expertise, their guidance, administrative and institutional support, and the funding that makes our work possible. So thank you to everyone involved. And with that, we will wrap up this introductory session. We're going to now learn about some of the global and collaborative initiatives that are happening in TB vaccine R&D. And I'll turn it over to our first speaker, who is Rasmus Mortensen.
Thank you so much, Jennifer, and thank you for giving me the opportunity to just speak quite quickly to you guys. I want to echo Morten in the first session. Thank you all for being here in Copenhagen. It's really exciting to see so many TV people in Copenhagen. So thanks for that. I'll just take a short while to explain to you a little more about NIBI, because NIBI is kind of part of the reason why we're sitting in this room also here. And we typically get the question, what is it actually you're doing there? So I'll spend just 3, 4, maybe 5 minutes explaining to you a little bit what it is and what it's not. So NIBI is quite exciting, especially in these times where funding is scarce in this space. It's the Novo Nordisk Foundation Initiative for Vaccines and Immunity. And really, it is a two-pronged mission that we have in the center, where I'm fortunate to be the PI of the TB part of the program. First mission is really a key thing when working with any different vaccine or any diseases, to generate knowledge on host immunity and host pathogen interaction and vaccine technologies. So these are really enabling sciences that allow us to focus on point number 2, which I think is really one of the unique things about NIVI compared to any other research program, and that is translational science, which will be covering the whole space from early discovery and innovation to development and clinical science. So the second part of the mission is really to translate all these findings and insights we have into vaccines that provide robust, durable, and broad immunity against respiratory pathogens. And to reach this goal, we are building, expanding, and strengthening research infrastructure, primarily in the Copenhagen area. And the way it's going to be built is there's going to be different groups, different areas of research and development coming together to solve these or to achieve this mission. And there'll be groups with different PIs working, for instance, on the targeted respiratory pathogens, where to begin with, we're going to focus on 3 pathogens, where TB is one of them, group A, strep and influenza. And the idea behind selecting these 3 pathogens is really there's a lot of synergy between these 3 pathogens, so you can learn from one and use that knowledge in other. But it's also representing upper and deeper airway infections to cover, in the incidence of a new pandemic, we will gain insights on both immunity in the upper and lower airway. There'll be groups focusing exclusively on airway immunity, sampling and analysis of samples. There'll be vaccine platforms, where there'll be a portfolio of platforms where we can do head-to-head comparisons, both preclinically and clinically, really a unique aspect as well to allow us to do these head-to-head studies. There will be people working on formulation and airway delivery, both in terms of priming, but also systemic prime and airway pull. I think I said already that there'll be assessments of airway and systemic immunity, both preclinically and clinically. And how to achieve this is part of the engine and the design is really to have 2 components of this initiative, or center if you want. There'll be an NIVI Research Center localized both here at the University of Copenhagen, but also a hub at Statens Serum Institute, where I spend most of my time, where there'll be the research engine and the discovery and education part is centered here. And then there'll be a development component, which is a private entity, will be the translational capacity you wouldn't normally find in these types of research settings, where there'll be vaccine development capacity, there'll be clinical trialists and clinicians, but also IP management and business development capacity that will enable us to translate all the findings from the research into development and hopefully in the end products that will go to the market. And all of this is made possible through a fairly generous grant from the Lloyds Foundation of about these 240 million committed for the first 8 years of the lifetime of this initiative. So physically now you are in Copenhagen, so now you can see how it's all organized. We're sitting in one of the components of the research, the main component of the research center here at University of Copenhagen, where we have the NIVI hub. It's part of an existing leading clinical research and educational environment. We have on-site state-of-the-art research infrastructure. And then in a very bikeable distance from here, you find SSI, where I've spent again most of my time. It'll be a partner hub where we have relevant research groups covering adjuvant research and TB research, where I am, but also an on-site BSL-3 facility where we can do the TB work. Of course, the idea is to strengthen and expand on this research capacity here in Copenhagen, but we'll know that we'll not be able to do everything here. So a big component of our work will also be to engage with both national and international partnerships. That goes without saying, and that's of course why this is also relevant to the audience here. So finally, just a brief word on what we're actually going to do in the TB program where I'm the PI. The 3 goals that I've listed for the first period of this is to define key mechanisms of protection and pathology in the lung. And I think the pathology component of this is really important when we also start to think about immunizing in the airways, which is a very delicate tissue that we want to protect. And we'll have an initial focus on unique aspects that are not covered so well at the moment, focusing on Th17 cells and tertiary lymphoid structures that are also emerging as very important for other airway diseases. The second one is huge, and we want to identify biomarkers of lung immunity. See if we can find blood predictors and signals guiding kind of quote unquote lung seeding. if the cells that go to the lung are lucky or they're on purpose. We'll try to figure that out using both animal samples and samples from our ongoing and planned clinical trials with our TB vaccine candidates in the future. And finally, to use all this to develop next generation TB vaccine strategies for induction of airway immunity, both by IM delivery, but potentially also by immunizing in the airways directly. We hope the outcomes will be an enhanced understanding and contribute to the large field of researchers to enhance our understanding of vaccine-induced immunity to TB and synergize with the other priority pathogens, to bring forward the new vaccines and vaccine concepts for lung immunity in general, and to identify and validate biomarkers to support and accelerate the development of our own candidates, but of course also other vaccines in the field and synergize with the already existing fields of scientists. I think with that, I'll just say I'm really excited about this and looking forward to keep you updated on this in the future. Thank you so much.
Thanks, Rasmus. Exciting things happening here in Copenhagen. Our next presentation will be by Ellie Van Wright from TBVI. She unfortunately could not be here with us in person today, but they are doing some great work with the product development pathway that we wanted to share with you. So she's done that through a recording.
Sorry, we can't hear the presenter. We are just seeing the slides, but the presenter we can't hear. Thank you.
Can I hear?
Hi, we are still not able to hear the presenter. Kindly.
We still cannot hear anything.
So thanks to Ellie, and I apologize for those online. We are trying to figure out what the issue is with the audio online, but we will place the presentations online so that you will be able to watch them after the session. And our apologies again, we are trying to address it. We are aware it is an issue. The next presentation is also recorded, and again, we will have this online if we're not able to address the audio issues. Frank Koblenz, who also couldn't be with us today, provided a presentation on a progress report they are doing on the TB vaccine R&D roadmap that was launched 5 years ago. So that was, apologies again to those of you online. That was our last recording. The rest you should be able to hear. Our next speaker is Geeta Gersing, who's going to give us an update on the accelerator and the SAGE.
See if I can line up this. Thank you very much. Good afternoon, everybody. I've got just a few minutes to give you an update on the TB vaccine accelerator. This is the third time that I'm presenting in this conference to this group on this. The first time in 2023, this was very sort of conceptual. I think last year we talked about the things we were going to do, and then this year we've got quite a few updates. I'll be quick, and you please do follow up if you've got any more specific in-depth questions. So, for those who don't know, the TB Vaccine Accelerator was set up in 2023. It aims to expedite progress towards development, regulatory approval, but really importantly also access to new TB vaccines, and specifically those for adults and adolescents. It's really conceptualized around technical and strategic working groups. The council meets once a year, and that's really formed by ministers of health, civil society, and donors. So I'm going to give you just a quick update on 3 of the working groups where we have been actually advancing through some activity. And the first is under the Product Development, Manufacturing and Policy Working Group. This is a technical advisory group for WHO that was set up at the beginning of this year, really focusing on clinical and policy considerations. And there's been some discussion already today about pre-qualification and how that really informs uptake in countries. For vaccines, there is also a strategic advisory group of experts that needs to give a policy recommendation. And so Saskia Denbon, who is in here in the audience, has been leading a group of experts towards actually having the first real in-depth discussion with SAGE that happened in September of this year on essentially the data that we anticipate will come out of phase 3 clinical trials. So looking at the pipeline, obviously M72 is the most advanced, but actually there are a lot of similarities between different vaccines that are also coming through that pipeline in terms of their clinical development and pivotal clinical strategy. So we got our first initial feedback from SAGE. There was some very sort of helpful outputs, such as that acknowledgement that we are not going to get efficacy in every country that is included in the phase 3 clinical study. It's likely to only be powered from 1 or 2, but that's okay as long as there are not huge epidemiological differences between countries. There's also the acknowledgement from SAGE that they will not see efficacy in IGRA-negative individuals, that all of the trials currently are powered to show efficacy in IGRA positive, so that's something that they really need to get their head around. So we're working on that. They gave us a recommendation to go away and look at scenarios of clinical trial outcomes and to plan around what the risks and potential mitigation strategies would be now, so that we can inform them, but also generate any data so that we're ready for a policy recommendation. We actually had a TAG meeting this morning on that very topic. And then lastly, we've been starting the discussion on the inclusion of asymptomatic TB as a potential clinical endpoint, and we'll be having follow-on discussions with regulators on that next year. So that's on working group 2 on the clinical and policy. We have also been working very intensely on working group 3, and this is to do with finance and access. There was an accelerator working group that was set up at the beginning of 2025 that has been co-led by the WHO, Gavi, and the government of South Africa. This has been an incredibly intensive amount of work looking at what are some of the licensing agreements, IP and access provisions of the candidates in the pipeline. What are some of the anticipated needs of countries to introduce and roll out those vaccines? Looking at a different range of countries. Gavi supported early introducing countries. Looking at whether countries are intending to self-finance, or how are they going to pay for these vaccines if and when they become available. And then finally looking at 10-year supply and demand forecasts to really inform vaccine developers about what they need in terms of scale. But most importantly has really been this report that was launched 2 weeks ago at the G20 looking at potential novel financing solutions for new TB vaccines, acknowledging that there are some countries that are supported by Gavi, but actually the majority of the burden is in middle-income countries who will need to think about how to actually finance and procure these vaccines. So this work is now moving forward with implementation of some of these financing solutions in the next couple of years, so that hopefully we have some mechanisms in place when the first trial efficacy results start to come through. And then just quickly hopping now across the continuum to working group 4, we've been doing some work through country consultations, and the last time that we had this meeting, we had just closed down a meeting in Indonesia, working with various stakeholders, looking at things like what kind of data and evidence are needed for decision making, what can we learn from other vaccines that have been deployed or other countries that are thinking about deploying TB vaccines, and how do we start already thinking about a better engagement of the community to build trust around these vaccines. There was a report that's been published, but actually very recently now in July, we had a very similar meeting in South Africa with some of the same aims, really trying to understand what's needed to get ready to generate evidence and get everybody on the same page. This was co-convened by WHO and the Department of Health with support from the Orem Institute and funding from the Gates Foundation. So that report is now ready and is actually informing the development of a national roadmap for implementation of new vaccines in South Africa. That work is ongoing. But I think what we really wanted to tell you today is that we are now moving towards actually launching this working group 4 on country readiness, community partnership and advocacy. And rather than having a working group of experts, we're really trying to think about how to conceptualize this as a community of practice. This is still conceptual, but we are really basing it on the lessons we learned from the experiences we had in Indonesia and South Africa, where we are really seeking to understand from the global TB community what are the big questions, the big themes that we should be trying to convene around. And so the idea is that the TB Vaccine Accelerator proposes some cross-cutting themes or some deep dives in countries or sub-regions, as we've done previously, to the TB community. We seek feedback. We've already started some of those consultations. But we then actually use those conversations to prioritize the big questions that these convenings need to focus on, whether they be cross-cutting, whether they be country or multi-country specific. We then actually work with in-country partners or in-region partners to organize convenings, and we start to put together a steering committee to actually design the consultation, identify the important questions, and put the program together and bring the community together. So what we have proposed is that we will do 6 of these over the next 2 years. We don't have the themes for you yet. We're working on that. But I think similarly, Jennifer, to what you showed about the community that you're working with for this working group, we are envisaging something similar in terms of being able to access materials and share what we learn. So that's it from me. Just really a shout out also to the Gates Foundation and the Wellcome Trust, who are now funding this work going forward. Huge thanks for their support, and thanks to all of you.
Thank you, Ita. And then finally, we have Sean Palmer, who's going to update us on some workshops that have been held on funding for late-stage trials, something we all know is a critical issue.
Thanks, Jennifer. Hi, everyone. Sean Palmer from IAVI and the TB vaccine advocacy roadmap here. So I've just got 2 slides, and I'll try and keep it brief. And so this work has been kind of ongoing for the last 2 to 3 years, really reflecting on the need for sustained and diversified funding for TB vaccine research and development. Really facing the same similar barriers across the field, particularly for property-related and neglected disease innovation in general, and recognizing that the current funding system remains too fragmented and too insufficient, really depending on quite disparate grant funding, and the need to have more sustained, seamless funding to provide more long-term capacity for the field, but also making sure that there aren't long and costly delays in between trial phases, particularly when we reach late stage trials and the costs, of course, increase. And so in this context, we see TB vaccines as really highly relevant case study for how we could perhaps work towards more innovative and long-term sustained funding for product development at large. And through this, we developed a series of T20 policy briefs over the last few years where we've really conceptualized the thinking here and identified some key thematic domains that we wanted to explore as we take this forward to more concrete proposals. And so for this, we are really trying to push for funding to be more based on countries' national income, but also their research capacities and TB burden, looking at how can we leverage diversified funding from middle-income country funders as well, and look at funding or resourcing more broadly, not just monetary support, but also thinking around things like trial capacity and whatnot, to see how we can bring in new funders and ensure that access is also built into these funding mechanisms. And so I think these themes that you'll see on the screen here are very familiar. They also, I think, echo a lot of the work coming out of the Accelerated Council, and hopefully as well show a lot of opportunity to also increase funding in the research and development stuff to ultimately help drive ownership and engagement as we move through to implementation as well, so that the engagement of middle-income countries with high TB burden in particular can really support TB vaccine development end to end. And so over the last year, IAVI has led a series of workshops with global and regional stakeholders from including traditional high-income country funders, as well as those from middle-income countries, to really explore innovative financing strategies for late-stage TB vaccine R&D and where opportunities to bring in new and existing funders together in a joint and coordinated way. So this kicked off last year at the World Health Summit in October 2024, then at the Regional World Health Summit in New Delhi in India back in April, and recently at the 12th EDCTP Forum in Kigali in June. And so I just wanted to share just a couple of the high-level kind of takeaways here. All the reports are here if you want to go into more detail. But as you can imagine, one of the key points to emerge from these engagements so far has really been about the need to enhance the role of high-burden middle-income countries in vaccine financing for the development of vaccines by engaging early with diverse regional actors, strengthening the evidence base to address local and regional priorities, and make sure that country funders have the evidence they need to invest. And this works both for implementation and R&D, of course, as well as defining conditions for support. So how can we ensure that any resources of funding a middle-income country funder, for example, puts forward will help result in them getting access to the vaccines in an equitable fashion when they're available. Secondly, to diversify and coordinate resourcing, we've been looking at various examples of R&D financing initiatives, both ones that are active as well as those that have been proposed in literature to help inform the proposal we're developing here. Diversifying funders and coordinating resourcing to maximize efficiency and accountability as well by leveraging these existing models and networks alongside novel approaches. And then thirdly, to align the interests of different stakeholders and ensure robust coordination and leadership to support a whole system approach. So really looking there for synergies of where can we invest in TB vaccine development in a way that will help the broader research ecosystem, particularly the vaccine research ecosystem in TB endemic regions, ultimately then hoping to benefit the broader vaccine development capacities of these regions at large. So as next steps, we are going to be doing some more concrete or targeted engagement with stakeholders, both in the funding space as well as with product developers, really to see what are some of the concrete recommendations that we need to bring forward into an actionable proposal to hopefully drive forward more sustained coordinated funding for TB vaccine development. We recognize as well that the first vaccines to cross the finish line won't be the last that will be needed. We'll need to continue to innovate and have more effective, more accessible vaccines moving forward. So we really see this as an initiative that can support broader TB vaccine development beyond just those currently stage candidates. I'll be here all week as well, so if you want to talk more about this particular initiative, please do reach out. Thanks, Jennifer.
Thank you to our first set of speakers. We're going to transition to our next group, so you guys are excused. So our next session is around progress and opportunities in TB vaccine R&D. We're going to start with the clinical research with Alana Mumra from IAVI.
Thank you. Hello, everyone. Very happy to be here. Made it on time. Shoo. So looking a bit wrinkled because I came directly from the airport. But I'll give you an overview of what we are currently looking at in clinical development of our vaccine. So I'm showing you this slide. It's different to the one that Kelly showed. These are the candidates in actual active clinical trials. But my task today is not to talk about the candidates in development, because this will be very expertly done by Willem in a session on Friday morning. And I will focus on some of the other elements that you'll also see on the pipeline slide. And just take note that there will be these green boxes on my slides, and these are just to highlight the relevant TB vaccine sessions. And there are quite a few, which is very exciting at this year's union. So these are the topics that I'll be talking about. Some of it has already been raised in earlier talks. But really just to give you a very high-level overview of the current considerations in TB vaccine clinical trials. Prevention of clinical TB in adolescents and adults remains the most important target for global health impact. And according to the WHO PPC, it requires at least a vaccine with 50% efficacy against clinical TB. This is not to say that development of an infant vaccine and prevention of disease in infants is not important. This just shows that the development of an adult and adolescent vaccine will have a greater impact on TB incidence through the prevention of transmission. Prevention of symptomatic bacteriologically confirmed pulmonary TB disease remains the primary endpoint of choice or preferred primary endpoint. And of course, this requires participants to present with a TB symptom, at least one. And then also what we are seeing in the late stage trials currently is a more stringent primary endpoint that requires at least 2 positive diagnostic tests, which is a culture and an expert or any combination of the two. And then, of course, the sputum samples have to be collected before the participant starts on TB treatment. In the context of this endpoint, these trials have to be conducted in high incidence groups, in high burden countries, and in high incidence ages in high burden countries. They are then, of course, large, expensive trials with long follow-up, and unfortunately, this will be the reality until we have validated immune correlates of protection. So a question that is being asked by developers is whether the past pre-proof of concept trials with a prevention of infection or prevention of recurrence endpoint should still be considered suitable alternative endpoints, and whether they should be used as stage gates. And in the light of recent negative findings and some further thinking on this, it seems that this is not the way to go. And Ellie also highlighted this in her talk on the TB vaccine pathway, development pathway, so you're welcome to also go and look at that for more information. So the big question that we're all talking about and thinking about, and Gita mentioned it as well, is prevention of asymptomatic TB disease. So asymptomatic TB has been shown to be at least 50% of prevalent TB disease globally, and in more recent studies, actually as high as 80%, depending on whether you screen for symptoms and with an X-ray compared to whether you screen with an actual microbiological test as well. And we have standardized definitions for asymptomatic TB now. The relevant one here is bacteriologically confirmed TB disease without TB symptoms at screening. And in there is already an issue because the presentation of symptoms by a participant is not always that reliable. And then there are different considerations for trial endpoints. How do we bring asymptomatic TB disease into our trials as an endpoint? Some of the late stage trials are already having it in the design as an exploratory endpoint. And this could also be a secondary endpoint, and then compared to inclusion as a primary endpoint or a composite primary endpoint. This scenario could lead to potentially smaller or shorter trials, but you can risk missing out on a useful vaccine against symptomatic TB if vaccine efficacy is different for the candidate against asymptomatic TB and symptomatic TB.
a data gap remains, and we really need to urgently gather more information on the natural progression, transmission, and mobility of asymptomatic TB, and really to gather this evidence to be able to include it in a primary endpoint. And then there are of course various trial design considerations and thinking around how best to screen for asymptomatic TB at the start of your clinical trial. But I really encourage you to join these sessions on the slide for a really in-depth talk and discussion about asymptomatic TB. So our trials, our efficacy trials traditionally have restricted enrollment to IGRA positives or people who have a positive interferon gamma release assay as an indication of having been exposed or sensitized to MTB. These trials then lead to a higher TB case accrual and can be run with a smaller sample size. And the reason for the IGRA positive restriction is because IGRA negatives have been shown to be 8 times less likely to develop pulmonary TB. But recently, we have and our late-stage trials are doing this, we've seen that IGRA negatives are being included as a cohort in a late-stage trial for safety and immunogenicity assessments. And if we think about the question that Ether also raised, that was brought up by the SAGE, would this be enough if we don't have efficacy data in IGRA negatives? And here, there are some alternatives or options for gathering efficacy data. You could do the traditional type trial and power your trial for efficacy in IGRA negatives, but this would be even larger, more expensive, and much longer than a trial with IGRA positive participants. You could have a more novel mixed trial design, but these would have to be conducted in high MTB transmission settings, and this is based on recent modeling papers and exercises. And then the alternative is to, while you roll out your vaccine for IGRA positives, have a commitment to also deliver effectiveness data in IGRA negatives from participants or people who live in hotspot communities. So the real question is, will the country-specific regulators, will WHO SAGE allow use in both IGRA positives and IGRA negatives if we don't have the efficacy data? And this unrestricted initial policy recommendation, regardless of IGRA status, is uncertain. It could be possible that we could have a recommendation restricted to IGRA positives only, but then that would require IGRA screening, which would greatly limit global rollout. It's unlikely that we would have an unrestricted one. Most likely, would probably be a more conditional recommendation, as in the last example that I've mentioned, where you roll out the vaccine in IGRA positives, in everyone, but you have a commitment to measure in IGRA negatives for effectiveness during national vaccination campaigns. The goal really is to have this conditional recommendation or an IGRA blind SAGE recommendation. TB remains a major cause of morbidity and mortality in people living with HIV. TB prevention in people living with HIV through vaccination would really have a major population level impact. And currently we do see people living with HIV being enrolled, and these are well controlled, but people on ARTs, and they are included in trials where possible to gather at least adequate safety and immunogenicity data. They should also be enrolled in efficacy trials, and specifically for safety and immunogenicity, although your trial may not be powered for efficacy in this group, it could lead to a potential recommendation for use if the vaccine is approved. And then I think the most important thing is just to have a clear regulatory pathway and plans for trials to collect the necessary data for inclusion in the label. And ultimately, we want the vaccine to be accessible to people living with HIV, and this vaccine should be safe and effective, and the same as it would be for people without HIV. WHO has recently released this consensus statement where we are really urged to generate adequate evidence for pregnant and lactating women in TB vaccine research and development. And we should also try to collect data systemically, safety and immunogenicity data at least, and these could be done on women who are already enrolled in our late-stage trials, and this is being done in our late-stage trials currently. But ultimately, the goal is really to include women who are pregnant or breastfeeding women as early as possible, as early as feasible, as soon as the risk-benefit profile is favorable. So what can we do to get to a faster, smarter clinical development? We could have innovative and efficient trial designs with optimized endpoints. Importantly, endpoints should also be standardized as much as possible across trials to allow for comparability of data. we could enroll the participants regardless of baseline EGRA status, as in the mixed population example that I've mentioned earlier. We could include the prevention of asymptomatic TB disease in the primary endpoint. Of course, we need more information and final decision on all of this. And in trials should be conducted in targeted populations and epidemiology in high burden countries, in priority groups. I think Frank pointed out that there are also pros and cons and issues when it comes to high priority or priority groups. And then, of course, all late-stage trials should contribute samples towards a biobank to identify correlates of immune protection. And we already have on this ongoing program and efforts to identify a correlate or correlates based on samples collected in earlier trials. And then there are also ways to accelerate transition between clinical development phases. These could be seamless phase 1A, 1B designs, or possibly seamless phase 2-3 designs. And then lastly, I'll conclude, although I didn't speak about candidates in this talk, I want to emphasize that diversifying and refueling the clinical pipeline remains a priority. We have global and collaborative efforts to address these scientific and logistical challenges in clinical development, and these are ongoing, and the public-private partnerships here are key. And then also ensuring equitable distribution to new TB vaccines is a critical challenge. So as you can see, there's large focus on implementation research that we currently see. And then lastly, we can do everything right and we can make sure that our vaccines are accessible, but we need our communities to take up a new TB vaccine. And this is where I'll end my talk. And I think it leads nicely into our panel discussion that will come later, right?
Yep.
Thank you.
So on for that great summary of all that's happening in clinical research. We're going to jump right into the early stage and preclinical research. Unfortunately, we have a very tight timeline, so we aren't going to be able to take questions, but I will speak to our speakers. I'm sure they will be glad to answer your questions individually.
So I'm gonna go ahead, and I was sort of given this challenge of like, what was new this last year, of which there's a lot new. And I already apologized in advance because there's no IL-17 in my talk. I'm so sorry. But I'm gonna go ahead and so basically, oh, that's the wrong one. Do I have to go this way?
Middle one.
What?
Middle.
Oh, great. Okay. So I'm going to start. I just have a few selected areas that I wanted to cover. And the first is antigens, because I think we've heard a lot about how we need new antigens and the like. But I'm going to start by saying that we're all kind of here building on a lot of work from a number of giants, many of which are in the room and many of which were actually done here in Copenhagen. And this is just to point out that if we look at the work done by Peter Anderson and his group, and many of you guys are in the room who did this work, Steve Reed at IDRI, studying that with Tom Ottenhoff, looking at latency and hypoxemia antigen, all the way back to myself and Cecilia doing more genome-wide approaches. We know a lot about what are the antigens in TB. And so I think the questions are really not so much what's the world of those antigens, but how do they work? What's the context? So there's a couple of themes I just wanted to touch on. The first are those that might be associated with protection. I highlight, and I think this is a study that you probably heard about last year, but it's from Munya, Esposi, they said it from Politango and Joel Ernst, who really were able to describe a couple of antigens using TCRs from subjects in the adolescent cohort that didn't go on to get TB and therefore considered protective. The other thing I wanted to mention is I think we're beginning to hear about antigens in people who are resistors. I couldn't find anything that was public in that domain, but I think we'll be hearing more about that this year. The next thing to kind of really highlight is really this topic of what are the antigens that are really displayed by the TB infected cell. And I'm going to start with this. This is an old study, but this was a phase 1 study done in Portland. These are people who got BCG plus an adenoviral vaccine. And if you just look at the bottom here, you'd say, oh, they had fantastic responses. That's what we would typically do, is measure immune responses to the components of the vaccine. But when we went ahead and generated clones from those vaccinated donors and then tested them against the TB infected cell, the majority did not respond to TB at all. So this graphic down here is misleading. And this sort of highlights some really nice recent work. 2 papers actually from Brian Bryson and his team And what they've really done is to take TB infected cells, strip peptides from those cells, and to begin to characterize those peptides. And I think what Brian would say is they're not finding new antigens, but they're really beginning to understand like what's the basis for why those antigens are displayed in this particular kind of secretion system that they find. And then kind of parallel to that is some nice work that just came out of Steve Carpenter's lab, where he actually looked at T cells that either responded to TB or TB lysates and sort of did TCR sequencing on those, and again found that the CD4 T cells then were also had this common theme of the CSX7 secretion system. So just pointing out that understanding the context is likely really important. The other thing I would add to that we don't know is potentially which antigens are preferentially displayed in the places where TB is, that's the lung, granulomas, et cetera. So I think these are going to be themes over the next years. There's two really nice studies that are ongoing for RNA vaccines. The first is BioNTech, and this is the BNT164 vaccine that was included in the pipeline we just saw. This was data that was actually presented at Diablo Ray, and they made a nice multi-antigen construct for this. And what you can see is they induced very strong immune responses, both T cell and B cell responses. And when they were given either as a vaccine alone or actually following BCG, you can see that you actually got a decent protection in a mouse model. The other one is by Akagera. This is a company you probably haven't heard about yet. It's a company that's actually owned by the Rwandan government. And they're also doing RNA vaccines that have a targeting sequence that allows them to be targeted to different intracellular components. This particular one goes to endosomes, and you can see that it's better taken up by dendritic cells, that the RNA is better expressed, that when they make their particular construct, they can measure very robust CD4 and CD8 responses. And here they're taking advantage of the low dose model, which I'll describe in just a moment. But here the key outcome is actually the number of animals that didn't get infected. So it's really a prevention of infection output. And you can see that their vaccine, so where did I go here? This is the pointer. And now it's not working. Anyway, their vaccine, which is on the bottom right, 15 of the 20 animals didn't get infected at all. So this is really kind of a striking endpoint. They're hoping that their vaccine will get into early phase 1 trials sometime in the next year. Just to point out that we all kind of know that adjuvants are limiting. This is some nice work that actually comes from Ria Kohler's lab, where they've created a synthetic TLR4 agonist. And then the other one that I wanted to highlight is LMQ. This comes from a Swiss concern. And this is actually MPL with QS21. And this is a study actually out of Helen McShane's lab where they gave PPE15 along with this LMQ. And what you can see, I can't see, oh, there it is that the giving the PPE LMQ followed by Chadox with the same antigen was reasonably protected in the mouse model. I do want to call out the low-dose challenge model. So this comes from Kevin Ursulaw's lab. And he did this in such a way that about a third of the animals will get infected when challenged by aerosol. And so what you can measure here is the proportion of animals that don't get infected at all, which I showed out here. And so what you can see is that BCG vaccination results in a higher proportion of animals that don't get infected, at least in the short term, and a lower spread of it to the contralateral lung. And then finally, just to highlight the non-human primate progress, this is work from Joanne Flynn's lab. As you all know, IVBCG is a very efficacious vaccine. And what they did is deplete either CD4 T cells or CD8 T cells to sort of ask what was important. And interestingly, what they found is that was the both the CD4 T cells and the CD8 alpha T cells that were most protective. And this is actually a nice cartoon from Dan Barber, just hiding that CD8 alpha T cells are actually enriched for sort of non-traditional T cells. That is mate cells, NK cells, NKT cells, and other cells. So their argument, in a sense, is that those effectors, CD4 T cells and unconventional T cells, were best associated with IVBCG protection. And then finally, you've already heard, there's several nice studies already that have looked at the human challenge model, mostly in the context of BCG. The most recent one, actually a nice report from Helen McShane's lab. And then I think you've all heard about the triple kill switch strain that we're hoping to see move forward. There's a paper showing that at least it can be, can actually provide infection in animal models and appears to be safe. So I'm going to conclude then with this. And this is a picture that I stole from Nicole Fram, who's up there, who along with Elisa Nemes has been heading a fairly heroic effort to define correlates in the M72 vaccine array. And my argument is that for us to speed this process up, we need to do what I'm going to call experimental medicine. That is, it's imperative to actually figure out comprehensively what a vaccine can do before it gets into these late stage trials. And Nicole, that is her program right now, is sort of asking a number of different outputs to see what worked. And I'm just going to argue that we need to do that up front so that we know which things to look at when we get to immune correlates. I'm going to argue that there's almost certainly not one correlate. It's almost certainly going to be related to a specific vaccine. But I'm going to argue strongly that we need to have the kind of the will and the resources to fund those studies, even at the very early stages of a vaccine. So I think that's it. And thank you very much.
Thank you, Dave, for that overview of what's happening in the earlier stages of research. I'm going to invite our panel to come up. And we're going to conclude, as Alana mentioned in her presentation, the importance of preparing communities for the uptake of new TB vaccines. So we're going to end with a panel discussion on just that topic. And I'll turn it over to our panelists.
Yes, okay, that works. Okay, hi everybody. Very happy to be moderating this panel with three advocates. My name is Mike Frick. I co-direct the TB project at Treatment Action Group, where I mostly focus on advocacy for new TB vaccines. And today we've been charged with talking about how to prepare and engage communities to get ready for new TB vaccines. We don't have them yet, but they're coming, and the community work should start before they arrive. We have about 20-25 minutes, so I'm just going to get right into it. I told our panelists the intros would be short. They're all very esteemed, but I'm going to introduce each quickly, and then they can say more about themselves if they'd like to. We have Priyanka, who's with the Global Fund Advocates Network and longtime TB advocate. We have Peter, who leads an NGO and is on the Gavi CSO Civil Society constituency to the Gavi board, and Limakatso, who's at ARI in South Africa. And you are also coordinating some of the national conversations in South Africa about TB vaccine readiness. My first question is sort of recognizing the moment we're in. I think it's fair to say this is not a good historical moment to be working on vaccines. It's not easy. And as the American on the panel, I want to acknowledge that some of that hardship is coming from my country. But nevertheless, there are reasons for TB vaccines specifically to be hopeful. And part of that hope is anticipating the future. Thinking about what we need to do to make that future possible. So my first question is to you, Peter, and it's about in this moment where we're still 2 to 3 years away from a new TB vaccine at the closest. What do we need to be doing with communities now while the science is still underway to get ready and make the most of the time that we still have before we know results?
Thank you very much for that question, Mike. The community is very expectant, considering that the only vaccine that we have has been there for quite a long time. And so they are looking forward to a superior vaccine. And we are coming from a background that this was a vaccine that you were being given once as a baby in a lifetime, even though it was not effective during the whole of the lifetime. And so people are expecting that what we should be, we should be giving hope to the community. And giving that hope to the community means communicating with them on the new development, having community dialogues on the progress science is making. And also considering that we have had, we have just 3 years ago, we had the COVID vaccine that had some issues that were myths and mysteries about it. that push the community away from the issue of vaccine and the boosters and all that. So communication with the community is important. That's number one. Number two is the issue of the scientists talking in the same language, because we had scientists, anti-vaxxers that were there during the COVID, and the community really was getting confused on who is telling the truth. They gave all the reasons why the vaccine for COVID is not good. And the scientists also gave the reason why it's important that you get vaccinated to prevent hospitalization and to prevent progression of the disease. So it's important that that communication is done early in the early stage. And lastly, Mike, you have mentioned about the progress of science in the upcoming vaccine. They're not yet there, but we need to be preparing the community, considering that some of those vaccines are going to be for adolescents and adults. And this is a category of people, not like babies that the mothers carry them and they had no choice. These are people that we have to talk to early enough to convince them to take the vaccine. Otherwise, they will make a different choice. And that's the important thing about it. Thank you.
Thanks. I think bringing up adolescent and adult vaccination is a good segue to a question that I wanted to ask you at the end, which is that in South Africa, how have these conversations started to take place? I think you were involved in the workshop that Gita highlighted that took place about vaccine readiness in South Africa. What are some of the issues that are coming up in thinking about the important things to prepare for when thinking about how to get a new TB vaccine to South African communities and all of their diversity?
Yeah, no, thank you. So there has been several discussions, and really one of them, as Richard explained, it's about talking to the communities. Community engagement has been regarded as one of the important elements of getting ready for the launch of the new novel TB vaccines. But the second one has been about delivering the vaccines at the right place. If we put them in a place where they are not accessible, where it's not feasible, it might make it difficult for communities to access them. Our current expanded program on immunization, I know for South Africa, and I'm sure for many low and middle income countries, is not ideal for vaccine delivery that targets adolescents and adults. It's mainly designed for young children. But there could be possibilities to integrate that with other, because they have experiences in delivering vaccines on things like supply chain management and maintaining cold chain. They could be integrated into other programs to make sure that the vaccines get delivered to the right people. But there will still also be a need to strengthen some aspects of the EPI program, like pharmacovigilance, as well as pregnancy registers, to make sure we are ready to deliver these vaccines. And unless it's convenient for the people that need to receive it, unless we start thinking about hybrid models that includes maybe both community and health facilities delivery, and unless we start thinking of other innovative approaches to reach populations that are at high risk of developing TB, like men who are often not found in health facilities, we might also have challenges of uptake. And we know that if we have the wrong location or an inappropriate delivery system or platform, or have low uptake and a lot of vaccine wastage. So we need to start the dialogues now on where we need to deliver these vaccines. They've started in South Africa, and I hope many other countries will join in the conversation.
Let me ask you a follow-up. So you're focusing a lot on where to deliver the vaccines, but I'm also hearing you say to whom. You know, you called out men in particular. I feel like in some of these conversations about who to vaccinate, there are kind of two general approaches that come up. One is, let's go for a general population of adults and adolescents. do a big campaign. The others, let's go through targeted risk groups that we know are at high TB risk, people with HIV, pregnant women, if it's safe to give it to them, household contacts. What is that conversation sounding like in South Africa so far, that general versus targeted?
The conversation at the moment is that if vaccine is available and there are no issues about shortages or financing, let's reach as many people as quickly as possible, the general population, the pregnant people, the lactating mothers, the adolescents at schools. However, if there is shortages and concerns with vaccine supply or financing, we need to then look at how we can strategize to reach the people that need it the most, to have the most public health benefit with the little resources that we have.
And of course, the report that came out of the G20, South Africa's G20 last week, 2 weeks ago, that Geeta also mentioned, basically told us that based on the supply and demand forecasts that Accelerator Council working group put together, unless there's some kind of corrective intervention, at the moment, it looks like there will be a mismatch between supply and demand. So we won't be in this unconstrained scenario you described. We'll be in a place of having to make choices unless we figure that out. And so that brings me to you, Priyanka. How do we get the global structures organized to figure out this supply, access, financing component about making new TB vaccines available? You work a lot with Global Fund. I'd like you to talk a little bit about the role of the fund as you see it, and maybe how you think Global Fund should work with Gavi and other international institutions.
Thank you for that question, Mike. We know that, there will be challenges when it comes to the implementation level, but it is for the communities to be more proactive and to sort of get ahead of the curve in being able to not just lead the process, but also to be able to support organizations and the structures that exist within within organizations like the Global Fund. We are in a desperate time. We are in a time where the aid landscape is shrinking. So it's up to communities to not just think about the Global Fund and other organizations like the Global Fund in terms of a monolith, but also try and understand the inner workings and what are the different mechanisms within the Global Fund that can be used to be able to get preparedness on the ground right now. We are very, very close to the replenishment moment, which is going to happen on the 21st, and then we are stepping in to the Grant Cycle 8 very, very soon after that. But Grant Cycle 8, or the 3-year implementation period as we call it for the Global Fund, is not going to be the time where the vaccines are going to be available. So what is it that through the Global Fund mechanism or through the catalytic investment of the Global Fund can be done right now? Can we start talking to communities, as Peter said, about vaccines? Can we start bringing groups together to already build champions, advocates who have been at the forefront of other preventive mechanisms like the TPT, like TB preventive therapy, to also start thinking about what's the science of the vaccine, how many doses, who are going to be the people who are going to be in the first wave of implementation according to the history of the government programs in the country. How can they be roped in? What are the challenges? What are the misinformation and disinformation challenges that they have been facing? And specifically, also when working in the context of the Global Fund, learning through the other structures, learning from the rollout of the malaria vaccine, learning from the challenges that the malaria vaccine rollout has had, also looking at and reflecting at the challenges, as Peter said, in the COVID vaccine. Like, if there are dropouts between doses, why is that? And how can a community group not just get ahead of it, but have a strong base of community actors and champions on the ground who are ready and equipped with that knowledge, preferably currently in the Grant Cycle 8, that they are getting ahead of the curve of the anti-science misinformation, disinformation campaign that might hit us once the TB vaccines become available.
Great. We haven't had time today for questions from the audience, so I'm gonna give people an opportunity. If there is a question in the audience, think about it, raise your hand. I'm gonna go ahead and ask my next question and then I'll come back to you, giving you time. I'll come to you, I guess. But first, let me just ask each of you to quickly, maybe just give me a minute reflection. What do you see as the potential contribution of civil society and communities to new TB vaccines? What is the uniqueness of engaging these community structures? What can they contribute that a health system can't, that an INGO can't, that other partners can't? And I'll start with you this time, Priyanka.
I would have to say that vaccines don't save lives, vaccination saves lives. It's accountability and action-oriented messaging that gets people out of their homes and to the vaccination center, or open their doors and be okay with taking the vaccination. And civil society and especially community actors can be a huge support system there, where they can not only just lead by example, but also talk to their peers and motivate their peers in the language and in ways that are the most effective in communities, because they have not just the ground level understanding of the community context, but also understand at a very deep emotional level that this is my life, this is your life, this is your kid's life, this is your family's life. And that, I think, really adds the edge and brings the value that civil society and communities can bring.
Definitely. Yeah, a decision to get vaccinated has deep emotional and attitudinal roots, for sure.
Yeah, the civil society and communities can engage governments early enough. There isn't going to be enough money to procure enough vaccines for all the people that need them. And so the governments have to prepare to put aside some money for the novel vaccines. And the communities and civil society have that voice to get engaged in the budgeting, in the budget making process, and push for resources. Again, at the global platform, the civil society has been very vocal to supplement the voice of government in support, getting global institutions like the World Bank and others to help fund and also to implement and to raise funds due for the Gavi 6.0. So the society could be very strong in mobilizing resources, both domestic and global resources.
Following the money and mobilizing it. Nice.
Yeah. I think civil society can also contribute in encouraging government to move fast. It often takes an average of 17 years from the clinical trials being available to a new product being part of a routine practice. Unless civil society stands up and demands action from government, the effectiveness results will just be sitting in libraries.
Impatience. Yeah, totally. All right, we'll come to our friend in the front. I'm gonna run the mic to you so you can ask your question.
Just want to know from the perspective of community, if scientific development tell you that we have good data on those aged 50 years and up, then is it easier to push the community to do vaccination for those adolescents and up? Because we know increasing age will have increasing latent TB infection prevalence in Asian countries. So I have no doubt that even in African countries, it's still the same. So now the trial is only like 50-year-old. So how to, very difficult. Only adolescents to 50-year-old can vaccination. Is that much easier than if elderly included? Yeah.
Yeah, good question. The late stage studies are working in the 15 to 40, 44, 45-year-old age range. So let's think about people who are older than that. How do you see them involved in vaccine rollout and community work? Does it look different for older age groups?
I think the aim is to target all age groups. Of course, we know that TB incidence increases from 15 years and older, but data from South Africa, some of the diagnostic gap is in the older age group, in the above 50, where the missing number of people with TB is higher compared to the lower age group. So everybody who is at risk of developing TB disease should get vaccinated.
Again, at the age of 45 is the age where you have non-communications. That's where most of the people, as they grow old, they visit the primary healthcare for blood pressure and diabetes and all that. And so integrating a TB vaccination at that stage could be very convenient at the primary healthcare and bring the vaccine at the nearest point of care to encourage that age group.
And some older adults are eligible for more and more vaccines, right? Shingles, RSV. So in a way, it's this middle age group that is sort of the question mark. But Priyanka, anything to add to the conversation here?
No, also just to say that the older age group can also again be a big motivator, because especially in the Asian and the African context, we need to look at not just tailored messaging, but we also need to look at who is delivering the messaging, and this group can be the valuable voice in terms of motivating people because of the social position that they hold within the community. So another aspect to look at and another motivator to ensure that they are also brought into the fold.
I'm looking at Jennifer to see, do we have time for more? We do. More from the audience? Okay, I can't run the mic to you, but if you say it, we'll repeat it.
Many organizations need, especially as we think about a situation where we will have limited supply and need to prioritize. So governments need to prioritize what kind of support community organizations need in order to deliver those messages and be an effective community.
So for those online and anyone who couldn't hear, the question was, what support do community groups need to deliver messages to help get people vaccinated, especially if we're in a situation of limited resources? How do we support the community groups? Who wants to start?
Yeah, it's a difficult one. The easiest is to have simple messages that are easily understood to encourage people to get vaccinated. We saw that with COVID-19, the more we prioritized, when we were now trying to reach those groups that were not initially included, then they were not interested. So we need to be careful how we structure those messages, that they are simple and they are not discouraging the other groups or making them feel that they are not at risk of getting TB disease, so that they keep engaged and look forward to their turn.
Yeah, I think the other issue here is honesty. If we are honest with the community, that we don't have enough vaccines and therefore we have what we call priority groups, we have key population and we have at-risk population, then that community will be able to understand and say, okay, fine, I am not at risk to get that category of people vaccinated first if we don't have enough, so that I can follow up when there is enough vaccines.
I'd say most importantly, short-term funding immediately to be able to help the initial rollout. And the other thing is that oftentimes what civil society organizations and community groups struggle with is that because we are leading from a space of passion and we are leading from a space of self preservation as well to certain extent, because this is our community and these are our people that we are trying to protect. We get very action oriented, and our empirical data and our learning sort of get lost because we are such doers and not perhaps as great documenters. specifically reflecting on the example of COVID, where there was a lot of mistrust amongst the marginalized populations in some countries, because the distrust within the system already existed, and that sort of increased the barriers to access for people, because people were not interested in going out and getting the COVID vaccine because they didn't trust the system. Community experts, community champions, civil society know where those intersections lie. And it's very important for civil society groups to be able to have the kind of support to bring that evidence out. Community expertise and empirical evidence is evidence. And that's one juncture that can be really, really helpful, especially in early initiation, in early rollout, that we get ahead of the barrier of marginalized groups not being the only groups that are a part of the first rollout.
Funding, so critical. When you first said self-preservation, I thought you were going to talk about self-preservation of civil society itself, because it's a bad moment for vaccine work, and it's a really bad moment to be in civil society. I mean, civic space is shrinking in every country and region, according to the United Nations. Our groups and communities lost so much funding this year and are really operating on life support. So funding, funding, funding. I just want to echo what you said there. But also, I think I heard you say recognizing community data. Maybe part of that is setting up relationships between community partners and more academic partners to work together to kind of promote TB vaccines. I think that's worth exploring. Okay, one last question, or we're done. We can be done. Okay, closing remarks. Any final reflections from you three? I'll give you the last word.
Time for another question. I won't do closing remarks. If somebody else does.
We want to hear from you. You're on the panel. We'll close with the three of you, and then if people have questions, they'll find you over coffee. Yeah. And why don't we come this way this time? Just anything you would like to end on.
I think we should not act surprised when the effectiveness results for TB vaccines are out. Let's start preparing now.
Out there, the community is very expectant of the new vaccines, and they want a vaccine that is superior. And we, of course, science also has to balance some things. But we as communities, we are there to help roll out the vaccine immediately it comes out. And we are ready to prepare the communities so that their expectations might not be fully met, but is met to some extent, I think.
I would say there is a very important immediate need to be able to keep community within the conversation, in the loop and on the table, and understand that perhaps communities don't speak in the same language that researchers and clinicians might, but we are very, very eager to know, understand, and sort of be prepared with you together.
Thank you. I'll turn things back to Jennifer. Thanks.
So I'm going to go ahead and close our session. I just wanted to thank our speakers and panelists for really fantastic talks. I think we're all around, and I can now hand this over to the new drugs working group, who I just saw them.
We have a very quick 5-minute bio break, but please, we're on a tight timeline, so the next one will be starting in 5 minutes. Thank you, everyone.